Let us take a closer look at the basic structure of a cell using a microscope. We will look at a simple plant cell, the onion peel. Take one piece of onion and pull out the thin, transparent layer from its inner surface with the help of forceps. This layer is called the onion peel. Place the peel in a petri dish containing a few drops of safranin, red-colored stain, for 30 seconds. This will give a pinkish color to the cells and help us see them clearly. With the help of a thin brush, transfer the onion peel to another petri dish containing water to rinse the peel and remove extra stains. Now, carefully place the stained onion peel on the glass slide using a thin brush, ensuring it does not break or fold. Put a drop of glycerin over the onion peel on the slide. The glycerin will prevent drying of the cells and improve clarity for better visualization of cells. Slowly place a cover slip over the peel using a needle, such that no air bubbles get trapped. Use blotting paper to gently wipe off any extra glycerin around the edges of the cover slip. View the slide under a microscope or a foldscope. You will observe nearly rectangular structures under the microscope. These are the cells of the onion peel, which are closely arranged without any space between them. Try to observe the peels of the leaves of different plants around you. You will find that all plants are made up of cells.